Hello, and thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator at Simulations Plus. I'll be hosting today's webinar behind the scenes, making sure we don't have any technical difficulties. In this webinar, Dr. Revathi Chapa, Senior Scientist of PBPK Solutions, and Suvarchala Avvari, Scientist 2 of PBPK Solutions, and Susie Zhang, Vice President of Regulatory Strategies, will explore the complexities of transporter-mediated DDIs, highlighting cross-validation of multiple compounds for assessing and predicting their risk. Our Business Development Director, Andy Mueller, will serve as our moderator today. A few housekeeping items before we begin. We take your privacy rights seriously. By registering for and attending this event or participating in the Q&A session, you're allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. We'll address all questions in the Q&A after the presentation. And now I'll hand it over to Andy to introduce our speakers.
Thank you very much, Jasmine. Welcome to today's webinar, Mastering DDI Risk Assessment, Navigating the Complexities of Transporter-mediated DDIs. This is our third in a series of PBPK-focused DDI webinars that are being offered by Simulations Plus, and I thank you very much for taking time out of your busy schedules and joining us today. I'm Andy Miller. I'll serve as your moderator. I currently serve as the Director of Business Development for PBPK Services, QST Services, and Regulatory Strategy Services here at Simulations Plus. I'm pleased to be joined today by my colleagues Revathi Chapa, Suvarchala Avvari, and of course, Susie Zhang. Susie brings us nearly 13 years of experience directly from the FDA, in which she focused primarily on PBPK modeling and clinical pharmacology. During her time at the FDA, Susie oversaw and reviewed many PBPK DDI submissions, which truly makes her an expert in what the agency expects in terms of DDI investigation within the development pipeline. Revathi Chapa is one of our senior scientists and is heavily involved in the development of PBPK models for various applications, such as first-in-human predictions, food effect, PK in special populations, drug-drug interactions, and the list goes on and on from there. Revathi received dual master's degrees in pharmaceutics and pharmacology and toxicology from the University of Washington and the National Institute of Pharmaceutical Education and Research in Punjab, India. Prior to joining us in 2019, she worked as a research scientist at the University of Washington, where she collaborated with both the NIH and the FDA for the development of PBPK models to predict adult and pediatric drug disposition. Suvarchala is one of our DDI modeling experts here at Simulations Plus. She's been with us now for approximately 4 years and has focused most of her efforts on developing and validating PBPK models. Suvarchala has extensive experience supporting companies with their drug development programs in all stages of development. I've prepared a series of questions for Revathi and Suvarchala. While they take the time to respond to my inquiries, I welcome you to post your own questions in the chat. We'll try to answer essentially in real time, but we'll also reserve some time at the end of my questioning to allow for your Q&A. Revathi, if you would be so kind, please feel free to get the webinar underway and provide a quick overview of DDIs.
Thank you, Andy. Welcome, everyone. Okay, so in this presentation, we will be learning the importance of PBPK modeling for various applications of new drug applications. So here is a snapshot tool of PBPK, how systemic factors along with drug-specific factors are integrated into a mechanistic equations to describe various processes that a drug undergoes in the body, be it right from absorption or distribution or its metabolism or it's eventually the elimination from the body. So this PBPK tool integrates various factors usually coming from meta-analysis of information from various populations, and those factors are actually integrated into the platform, along with where if any new drug comes and we incorporate the drug specific factors into those mechanistic equations. So PBPK has been widely used for various applications in the recent times, and the main highlighted ones here are the prospective predictions in healthy and diseased populations, sometimes predicting the PK in special populations like geriatrics, pediatrics, and pregnancy. So we also can try to understand the formulation or dosing route or food effect with the help of PBPK modeling. The major application that stands out is drug-drug interactions. So if we look at the distribution of PBPK model application areas over the years, as you can see from on the left, over the period from 2013 to 2023, the number of PBPK applications that were submitted for FDA that involve investigational new drugs have been rapidly increasing, and there is more and more confidence built over the time. So if we dissect the area and see which areas are mostly used in terms of PBPK applications, the ones that stand out are the DDIs. So DDIs, especially the enzymatic mediated drug-drug interactions, occupy 57% of the applications that have been applied over years. And recently, in the recent times, the transporter-mediated drug-drug interactions are also gaining importance. As you can see, 20% of the applications occupy related to transporter-mediated drug-drug interactions. So again, this is again the snapshot tool, snapshot explaining the NDA submissions, the EMA submissions, and how what kind of models are being used with, especially with respect to transporters, whether it is for PGP, the models like digoxin, dabigatran are used. For OATP1B, the statins are majorly used, and for OCTs and MATES, the metformin is commonly used. Okay, so why do we? What is the relevance of drug-drug interactions? Why is it occupying a significant chunk of application area? So the drug-drug interactions occur when two or more drugs interact with each other. Since people have comorbidities existing together, they not only take one medication. Sometimes they have to take multiple medications, and whether they can be co-administered or not, it can lead to significant adverse effect or a subtherapeutic response. So these interactions can result in pharmacological or clinical response that actually differs from the response of each drug if they are taken together. So let's take, for example, there is a drug A and drug B. So drug B can alter any of the specific mechanism of drug A in the body and cause an increase in the systemic concentrations of drug A, so which can lead to toxicity issues, giving a concern for the compound. And then in the second case, consider a compound, drug A and drug C. Here, the drug C kind of activates enzymes which can increase the elimination pathway of drug A, and it can lead to a decreased systemic concentrations, leading to subtherapeutic response. So it is very important when when drugs are co-administered to monitor the effect, the final clinical effect. Okay, so let's take one example of this case of Suvarchala in 2001. So as many of most are aware that Lipo-Bay was launched in 1997 and 1998. So this compound is metabolized by 3A4 and 2C8. There is a minimal metabolism by 3A4 and 2C8 because when a itraconazole is administered with Suvarchala, it only changed the AUC by 15%. However, this drug is eventually removed from the market because of high risk of muscle toxicity and rhabdomyolysis. So For example, when these statins are taken with a compound like zenfibrozil, which can increase the risk of increase the plasma concentrations of these statins, sometimes it can lead to a risk of around 200-fold and cause a severe toxicity. So, so as you can see, this here a snapshot how the servistatin withdrawal affected the stock price of Bayer over that year. So it is very important to monitor for these drug-drug interactions. Okay, so what is the definition of drug interaction, DDI interaction potential? So here there are two terms that are more specific to understanding the DDI interaction potential. One is victim or substrates, which are synonymous. Here we refer to a moiety whose exposure changes due to inhibition or induction of an enzyme or transporter by a perpetrating moiety. So perpetrator is a compound, is a moiety that can induce or inhibit an enzyme or transporter. So the victim or substrate is the compound that is being affected, and the perpetrator is the one which is causing that effect. So FDA has many regulatory agencies have given the classification of this effect, whether it in terms of inhibition or induction. So there are three terms that are generally used, ranging from strong, moderate, and weak, where the impact on the plasma concentrations are strong. Like, for example, if the perpetrator causes more than 5-fold increase in the exposure of a sensitive substrate, it's a strong inhibition effect. Similarly, if the compound's exposure decreases more than 80%, it's a strong induction effect. So again, for moderate, if the change in AUC is around between 2 to 5-fold, it is considered to be a moderate effect. And for induction, it is between 50 to 80% decrease in the exposure of sensitive substrate is a moderate effect. And then the weak effect, where the effect is only between 1.25 to less than 2-fold, and anywhere between 20 to 50% decrease in the AUC of sensitive index substrate. These classifications especially stands out with respect to enzymes, because the interactions are strongly defined, and we have more specific enzyme-specific inhibitors that kind of help us to understand the impact on the systemic concentrations. However, for transporters, due to lack of specific inhibitors, this classification is still under research. So it's there could be compounds that could cause a moderate to strong effect, but because of lack of specific inhibitors, the classification doesn't apply to transporters. Coming to the interactions, for example, a perpetrator can interact with the substrate in different ways. For example, it can cause a competitive or reversible inhibition, where the effect ceases over time, and then there is time-dependent inhibition, where the compound actually degrades that enzyme and causes the degradation. And in some cases, it can also there is also induction, where the perpetrator can induce certain enzymes or transporters and cause the decrease in the concentration. So the induction with respect to transporters is still under the research is ongoing. There's only studies with respect to PGP induction, since it is co-regulated by PXR pathway, but other induction pathways are still under research. Okay, so coming to the guidances. So we have over the time, there are different guidances that have been developed. For example, if we look at from FDA perspective or EMA perspective, they kind of set rules on how to evaluate the DDI potential of interaction, DDI potential of new investigational drug. So these guidance are specific with respect to understand the drug in context, the intended patient population, and the therapeutic response. But over the time, with the effort of academia, regulatory agencies, and the industry, there's a more harmonized guideline, which is ICH, which is M12 ICH M12 guidance, which kind of lays out both the set of in vitro or clinical studies that needs to be done when a new drug application is filed. So if we look at the ICH M12, it suggests the evaluation of DDI potential of a new chemical entity mediated by following transporters. So right now, it suggests to evaluate PGP, BCRP, OATP 1B1, 1B3, OAT 1 and 3, and OCT 2, and the MATE 1 and 2K. Okay, so next, these are the transporter-based PBPK models. So if we look at the application of how GastroPlus software has been used in developing various PBPK models, here are some of the examples that are highlighted in green, where at substrate like substrates of PGP like edoxaban was studied to understand the PGP impact on absorption using GastroPlus software. And there is another compound, cefadroxil, where the PET1 is involved in the absorption, and the impact of PET1 was studied. Similarly, there are other cases which highlight how the GastroPlus software has been used for developing the PBPK models. And then here is a snapshot of some of the applications that were filed with either EMA or FDA. So here is one case example of trametinib, where this compound is identified to be a weak BCRP inhibitor in vitro, and also according to the cutoff criteria, it satisfied, and the PBPK modeling was developed and using GastroPlus, and had studied the evaluate the effect of trametinib on BCRP substrates like rosuvastatin, and see how much the impact could be. And eventually the EMA accepted the PBPK submission. And another case example is exitinib. It's also a intestinal transporter PGP inhibitor. Again, the effect of PGP on PGP substrates was studied and using GastroPlus, and this application was also accepted by FDA. So,
Excellent.
do you
Thank you for that, Revathi. That was a fantastic overview of DDI modeling and simulation in general. Really appreciate you speaking to the current regulatory requirements and regulatory recommendations on DDI modeling and simulation, specifically looking at transporter-mediated DDIs. So in the previous webinar, we focused on CYP-mediated DDIs. In the very first webinar, we focused primarily on regulatory requirements and regulatory recommendations. So thank you for that great overview. Thank you for bringing everybody up to speed. The question that I have is, how is transporter-mediated DDI specifically incorporated into Simulations Plus scientifically and mechanistically? And it looks like we've got some equations teed up right here.
Yes. So in GastroPlus, we do have dynamic simulations, which entail all three mechanisms that were previously mentioned, like the competitive inhibition, time-dependent inhibition, and induction. And the equation for the competitive inhibition does not only include just one substrate, but we can model multiple substrates of the given transporters. And at the same time, we can also model the effect of multiple inhibitors of a given transporter on the single substrate at the same time. Similarly, we can do the same for the time-dependent inhibition and induction. So we could study a matrix of compounds at the same time. The prerequisite for this dynamic simulations is we need to have either a full compartmental model or a full PBPK model for both your victim as well as your perpetrators. Next slide, Revathi. Okay, so this slide here is just a quick snapshot of the DDI interface in the GastroPlus legacy version. To give a quick overview of this DDI module, the left side of this DDI window displays the current compound, and this is the record which was opened on the main GastroPlus window at the time you are accessing the DDI module. And you have an option to select the designation of the current compound either as a perpetrator or victim. And the substrate and also we can do the steady state predictions and dynamic predictions in GastroPlus. For steady state predictions, we need to specify the metabolic profile, which is the FM and FG values, and these FM values can be calculated from the in vitro assays using the built-in converter within the GastroPlus, or can be entered manually by the user and then can be saved in the database. But if we have the KM and Vmax values already present in the database for this model, the program will use these values to calculate the FM values automatically. And if you see a small pie chart below this, below on the bottom side, there is this pie chart displays the metabolic profile in the gut and liver. And next to this pie chart where we have the substrate dosing, we need to have the time of the victim's administration relative to the perpetrator dosing and the simulation length, and these are all required for predictions both for the steady state and the dynamic predictions. Then coming on to the right side of the DDI window, we have an interacting compound, and this can be a record in the database that is open in the main GastroPlus window, or can be a different GastroPlus database. And by default, the database of the standard compounds that were supplied with the DDI module will open when we are accessing this DDI module. And the designation of this interacting compound as a victim or a perpetrator is automatically set depending on the user selection of the current compound. And you can see we have induction and inhibition constants for all the proteins that are enzymes or transporters for a given compound and its metabolites, which need to be specified for all predictions. And yeah, you can see that you can see a little column which says validated for each enzyme and transporter. So once the particular inhibition or an induction parameter for that enzyme or transporter is cross-validated against multiple substrates or perpetrators, we change the validation to true, which kind of giving us a message to the user, this interaction parameter, induction or an inhibition parameter is kind of cross-validated, and we have a confidence in that particular interaction parameter. Next slide, Revathi. Okay. So whenever a drug is taken through an oral route, it will travel through the different segments of the GI tract where it could be dissolved and some of the drug could be precipitated or degraded, and some would be exiting into the next compartment and so on. And the dissolved fraction, it travels through the gut wall where it could be facing some challenges with the enzymes that are present in the gut wall, where the drug could be cleaved into metabolites. So while it is traversing through the gut wall, there could be an uptake transporter which could aid in the absorption, like they help the drug to reach the systemic circulation. And also we'll have the efflux transporters that prevent the drug from getting into the systemic circulation. So these are the processes that happen simultaneously in each compartment of the GI tract. And whenever the two drugs are administered simultaneously, one drug could interfere with any of these mechanisms and can result in the drug-drug interaction. Next slide, please. Yeah. So in this slide, what we are seeing is the transporters of the clinical relevance, and you can see how transporters are distributed into different organs like intestine, hepatocytes, kidney, and brain. There are in each organ, transporters are divided into both uptake and the efflux transporters, and the one that are highlighted in the red circles, these represents the ones which are of clinical relevance and which have been extensively studied over a period of time. Next slide, Revathi. Okay. So the key aspect of the oral administration for both victims and perpetrators is not just to do a back of napkin steady state calculations, but to actually follow the transporters that are involved, the potential for the enterohepatic circulation, gut metabolism contribution, its inhibition, and then both active and passive transport into the liver and then distribution into multiple tissues. So it is quite important to get the correct distribution characteristics and thereby the initial model correct in order to have a good DDI prediction for oral dosage forms.
Thank you,
slide.
Suvarchala. Oh, sorry.
I have a couple more slides, Sandy.
Oh,
Sorry.
my apologies.
So, and in this slide, what we are seeing is a snapshot of how we develop our models, which involves developing a DDI qualification matrix. And this qualification matrix includes a different range of PBPK models for probe substrates, inducers, and inhibitors. And all these models are in the various stages of validation with the information that's coming up and evolving over time. So we always try to incorporate the new mechanisms, and also we do refine the models periodically so that it reflects the current understanding of the drug DMPK. And the list that we are seeing here is a comprehensive list of models that we have in-house. That are frequently worked on and validated with the new information. And we are also working on continuously developing the new models based on the regulatory interest. Next slide, Revathi. Okay, so coming to the model development and verification approach that we follow specifically for victim or substrate compound versus the perpetrator compound, there are three different approaches that we usually take. One is the bottom up, and then middle out, and top down approach. Bottom up is where we initially rely on the physicochemical properties, the in vitro metabolism and in vitro transporter kinetics and preclinical data to have an initial predictions on the PK of the victim compound. So eventually we would need some mass balance study or DDI studies with inhibitors of the major elimination pathway, or to confirm the transporter pathway to identify or confirm the fraction metabolized and the fraction transported contributions for the victim compound. And we start out with the bottom approach, but eventually we walk towards the middle out approach, which involves using of some clinical data to refine those parameters. And then the top down approach is usually using all the data, mostly coming from the clinical data, to make extrapolations for predicting the PK of the other substrates or other perpetrators or the victims, or predicting the PK in the other populations. Similarly, for the perpetrator, it entails the same process or the same approach as we have taken for the victim compound, but here we are looking at the interaction parameters like the Ki, IC50, K-inact, or the induction parameters. So we're going to work on identifying the induction or the inhibition parameters that the perpetrator causes, and then we verify that mechanism using relevant substrate models. Next slide, Revathi. So now we have, as we have discussed the model development approach, then comes the model evaluation criteria for DDI assessment. So the current assessment of the DDI prediction success is based on whether the prediction falls within the twofold range of the observed data. And this strategy results, most of the time it results in a potential bias towards a successful prediction, especially at the lower interaction levels. So now we follow the guest criteria, which was laid out by Guest and his group, and which is being widely used in the recent times. So this as you can see, there is a procedure for calculation of the limit, the upper and the lower limits, which involves a calculation of the ratio observed, which is the exposure in presence of an inhibitor upon the exposure when it is given alone. And similarly, the inverse process applies for the induction mechanism, induction phenomenon. And then next slide, Revathi. So so until now we have discussed that we can do or perform the drug-drug interaction between any compounds, but all we need to perform the DDI is models. So we have already built the models for the most commonly used compounds for the DDIs, covering a wide variety of inducers, different categories of inhibitors like strong, moderate, and weak inhibitors and substrates. But one thing that I would like to highlight here that we are trying to do a cross validation. That is when we are about to validate a substrate model, we try to validate the model with as many different inhibitors and inducers as possible based on the clinical DDI data that's available. And the same comes to the perpetrator, whether it's an inducer, inhibitor, or inhibitor, always validation is done with a multiple different substrates depending on the clinical data that's available. And the networks that we are seeing in this slide is to highlight some of the networks that are available for different, in terms of how the models were built and cross-validated for different mechanisms. And this is something that we provide to our users of GastroPlus. Um, and these are not just the models that we can use. You can always build your own model and use that to perform the drug predictions. But we just want to highlight that these are the models that we already created and can be provided to users, so they don't have to keep redoing or building the same models that we already have in-house.
Okay, so now that we have covered some important basic concepts of drug-drug interactions, also touching upon the regulatory perspective and also the model building and validation approach and the evaluation criteria. So now we wanted to discuss two case examples. First case example we would be discussing is OATP 1B mediated transporter DDI. Here we would be looking at PBPK model development of a compound BCRP and OATP 1B1 and 1B3 substrate, which is rosuvastatin. And we are going to see how we have validated the drug interactions with rifampicin or zamifibrazil. So here is the table highlighting the key physicochemical biopharmaceutical parameters for rosuvastatin that we've used in GastroPlus. So most of the physicochemical parameters come from different reference sources, where the parameters have been studied. Coming to the metabolism and enzyme transporter parameters, we usually use the enzymes and transporter data that was identified in vitro. We take the KM values from the in vitro values if they are available, and then we eventually optimize Vmax to characterize the right contribution of the transporter or the metabolism pathway. So here are the list of transporters that we have incorporated, and you can see how the KM values are coming from different references, and then the optimization of Vmax was done. So one might be having a question like, since there are multiple transporters are involved in the disposition, whether into and the distribution of this compound. So how do we know how much each transporter is contributing to either the disposition or the elimination pathway? So for this, what we do is we try to see if there is any information available with respect to in vitro experimentation. For example, for this compound rosuvastatin, there are papers from Wang and Zhang et al., where they have studied the contribution of these transporters in the in vitro using either sandwich culture hepatocytes or the simple hepatocytes, and use specific inhibitors to identify the contributions of each of these transporters. So we initially start out with this bottom-up approach where we rely on the in vitro data, and then we see if these values kind of correspond, if we use a DDI study with inhibitors of this and see if we can reproduce the results of clinical study. So if we look at the enzymes and transporters involved in the disposition of rosuvastatin, so rosuvastatin actually has a pharmacological action site located in the liver. So before it gets into the liver, there is some metabolism or the efflux transport that is happening, where the BCRP kind of effluxes the rosuvastatin back into the lumen. So there is some minimal metabolism by UGT1A1, which kind of contributes for the lactone formation. And then there are OATP1B1, 1B3, OATP2B1, and NTCP transporters are involved in getting the drug into the liver. And there could be some recirculation of this rosuvastatin and lactone into the gut by efflux transporter at this bile site. Leading to the recirculation. And there could be some compound that could be getting into the urine through a transporter like OAT3, and again the BCRP at the apical side. In order to characterize how much each of this pathway is contributing, we rely on both in vitro experimentation and also on the clinical information. For example, in some cases, we might know how much of the compound is excreted as a parent in the urine and the metabolite in the urine. So that information kind of helps to understand how much this pathway contributes to the overall disposition. So here is the modeling strategy that we adopted for rosuvastatin. Initially, we developed a baseline model integrating all the physicochemical in silico in vitro parameters. And we used, for the model development, we started out with the IV infusion and then oral tablet, 5 milligram and 50 milligram, which covers the dose range. So once we developed the initial model, then we would verify it in other studies. For example, not just one study, we verify all the studies that are available in the public domain, which covers the entire dose range. Here the dose range is between 5 to 80 milligrams. So we used all of those studies for verifying our model. So yeah, while we are doing that, we are also making sure the contributions of the transporters are correct by verifying the drug-drug interactions with rifampicin or here in case of zemfibrozil. So rifampicin is known to be an inhibitor of OATP1B1, 1B3, 2B1, OAT3 and BCRP, whereas zemfibrozil is an inhibitor of OATP1B1, 1B3, NTCP, OAT3, and it also inhibits enzymes like 2C9 and 1A1, which are involved in the metabolism of rosuvastatin. So this way, we are making sure that we are not just relying on the information that is coming from the in vitro, but we are also verifying with the clinical DDI studies. Okay, so here is a snapshot of the PBPK model, the initial predictions for the IV infusion and the oral. As you can see, the plasma concentrations are reasonably predicted. And here the black line represents the amount of drug that is excreted in urine. And you can see the dotted represents the observed data, and which very well in line with the simulation is in line with the observed data. Similarly, for oral dose, as you can see, the model reasonably predicts the PK of 5 milligram dose. Here, the amount eliminated is around 5 to 7% in urine for oral dose, and we are rightly describing the data. So this is the initial baseline model, and these are the plots, goodness of fit plots showing observed versus predicted values for Cmax and AUC. As you can see, the most of the data are within the BE limit, and in some cases, all the data are within the twofold, because sometimes the variability could come from OATP1B1 polymorphism, and it could deviate from that BE criteria. So this is the initial development and validation of rosuvastatin. And then we verify the drug-drug interactions with the studies available for rifampicin and zemfibrozil. As you can see here, this represents the predicted versus observed Cmax ratios of rosuvastatin. The green points represents the DDI studies with rifampicin, and the orange represents the DDI study with the zenfibrozil. So as you can see, they are more within the closer to the line of unity and within the guest criteria. So these are some other examples of model validations for statins that we have done. So this is for pravastatins, where we have used DDI studies with rifampicin, zenfibrozil, itraconazole, fluconazole to characterize the disposition, the transporters involved, and the metabolism involved in the pravastatin. These are for Cmax and these are for the AUCs. Similarly, for atorvastatin, we had the Cmax and the AUC plots. We not only developed the model for the atorvastatin, we also studied the metabolites 2-hydroxy-atorvastatin and atorvastatin lactone. So these 2-hydroxy-atorvastatin is also an active metabolite contributing around 30% contributing for pharmacological action. So we also characterize that. So as you can see, more or less, we are within the twofold criterion for both the Cmax and AUCs. So now that we have developed these models, how do we use these models? So for example, if a new investigational drug is file or like is being developed, so if it has some inhibitions towards OATP1B1 or the BCRP in this case. So we can see the effect of the compound on the substrate. For example, the in vitro inhibition parameters of those investigational new drug might have been characterized in vitro, and we can use those KI inhibition parameters and see how it impacts the PK of these statins. In that way, we can study how much it could impact the exposure of these statins. So yeah.
Yeah. So moving on to the next case study, which is about the PBPK-mediated transporter-mediated DDIs. Next slide, Revathi. So here we are presenting the modeling strategy or the approach that we have taken to develop and verify the digoxin model. Starting with developing the baseline model, we started with collecting the physicochemical and the biopharmaceutical data along with the in vitro kinetic parameters and so on. Once we have the model built, the next step was to see if the model could reasonably predict the plasma concentrations in after an IV administration followed by an oral administrations. We always try to start with an IV dose to have a confidence in the distribution characteristics of this model, then followed by the absorption characteristics using the oral data. And once the model is reasonably predicting both the clinical IV and oral data, the next step was to verify the model in different conditions like the fasted and the fed state, the different ethnic populations, and then followed by verifying the percent contribution or the FT contributions of that compound by performing the DDIs with the clinical PGP inhibitors like itraconazole, quinidine, and rifampicin. Next slide, Revathi. So this is just an overview of all the literature data that we have collated in order to build the baseline model. So one thing with the transporter is this, along with PGP, this digoxin is also a substrate for different transporters like OATP4C1 in kidney and sodium potassium ATPase in muscle. So we started looking into the literature to collect the in vitro kinetics of these transporters, but for sodium potassium ATPase, we didn't have any in vitro value. So we started with an assumption that the KM would be more or less similar to the other influx transporter, which is OATP4C1 for this model. Next slide, Revathi. So we all know that digoxin is a prominent victim drug to investigate the PGP-mediated DDIs. So the primary route of elimination for digoxin is the renal elimination via glomerular filtration and active tubular secretion. And this this little picture on the left hand top corner shows the schematic view of the transporters that were involved in the absorption and disposition of digoxin in various organs like gut, liver, muscle, and kidney. And to start with, digoxin is metabolically stable in humans, and its absorption and disposition is and elimination is primarily through a transporter-mediated process, and the digoxin is substrate of PGP. So the apical efflux of digoxin in these organs, mainly the intestine, liver, and kidney, is mediated through PGP, and the basolateral uptake of digoxin in muscle is mediated by sodium-potassium ATPase. And in kidney, along with the PGP, we also have an influx transporter, which is OATP4C1. When it comes to the biliary excretion of digoxin, it is happening through a combination process through PGP and MDR3. So we have also incorporated the MDR3 transporter in this model. So, and on the right hand side, we have a goodness of fit plots for all the clinical PK data that's available. And we have the predicted versus observed Cmax and AUC, and we can see that more than 50% of the predictions are close to unity, and then more than 70% of these predictions are within the BE limits of 0.8 to 1.25, and all the predictions are within the twofold criteria. And then we also have the model validity against the existing clinical DDI studies. And as I've mentioned previously, we have performed the DDI predictions with rifampicin, quinidine, and itraconazole, the PGP inhibitors. And we can see that the DDI magnitude predictions, which is represented in the triangles and the circles here, are within the twofold error criteria, and and it also within the guessed criteria, which we have seen in the earlier slides. Next slide, please. So this slide is just showing the PBPK model development for digoxin, where we started with the clinical IV data, and the blue solid lines represents the predicted model predictions for IV doses and oral doses, and the squares represent the observed data, and we can see the model is reasonably predicting the plasma concentrations following IV and oral administration. And for the first three plots, the orange line and the orange squares that we are seeing is the observed and the predicted urinary amounts. And we can see with this current model with all the transporters defining the transporter mediated process, we have more or less a reasonable prediction of the urinary amounts. Next slide, please. So again, we also worked on developing the effects of anidine model, which is also a PGP substrate, probe substrate, and the goodness of fit plots and the model validation against the clinical data is kind of giving us a confidence in the model that this model is reliable enough to perform the DDI predictions with a clinical PGP inhibitors. Next slide, Revathi. So starting with what happens when an investigational drug is a victim drug or an inhibitor? What the guidance says us, what is the approach that we have to take? So starting up with a bottom-up approach using the in vitro metabolism, and then we end up with the clinical DDIs or the mass balance studies. So if your investigational drug is a victim drug, and we do have an in vitro metabolism data and then the transporter data from in vitro studies, we can test the DDI potential as a victim drug, and most likely based on which mechanism is prominent, whether it is a metabolism or the transporter pathways, the regulatory agency recommends doing a clinical DDI with a strong inhibitor or a mass balance study to get an initial estimate on the FTs and FMs by each of these pathways. And once we know the relative contribution of the enzymes in the transporters, what we have to do is we can further test the effect of either the moderate or weak inhibitors using the models in the DDI qualification matrix. And if your investigational drug is an inhibitor, what we do is using the in vitro Ki values, the R values are calculated, and if R values are higher than cutoff values, then what we use is we use the relevant substrates to test the effect of the investigational drug on the substrate. Um, for example, our compounds in our DDI matrix can be used. And so that way, whether the interaction is significant or not, a clinical study may still be required, or sometimes if the interaction is not significant enough, then a simple sensitivity analysis could suffice. And we can submit to the regulatory agencies based on the evidence that, okay, this is the possible impact that we can expect from this interaction studies. Next slide, really. So we always face some challenges in predicting the transporter-mediated DDIs, despite there is a considerable research that's going on in the area of transporters over the past decade. The challenges arise because there is some system information that is still lacking in respect to the transporters, starting with the translation from the in vitro to in vivo of the transporter kinetic parameters. So the results from the in vitro studies, they often do not accurately predict the in vivo outcomes, and this discrepancy arises because the in vitro models that we use, they cannot fully replicate the complexity of the human physiology. So for example, there is a huge variability in the expression of the transporters between in vitro systems and in vivo tissues. The other challenge is that the transporters, usually they have a larger impact on the local concentrations, which is often which is not easily measurable compared to the systemic concentrations which we usually measure. And because of the active transport, the predictability of the tissue distribution is affected, which necessitates the scalar values to recover the measured volume of distribution values. And there is always a room for either over or under prediction of this hepatobiliary and uptake clearance extrapolated using the in vitro data. And there is no accurate contribution of the transporter in rodents is available, which could result in discrepancy when we are scaling from preclinical species to humans. The other challenge that we face is most of the transporters, and especially PGP, interacts with a wide variety of drugs, making it difficult to predict which compounds will be substrates, inhibitors, or inducers. And the variability in the drug structure and function could also add to this complexity. And there is always this interplay of enzymes and transporters, like PGP is often co-regulated with other enzymes like CYP3A4, which complicates the prediction of the transporter-mediated DDIs. And the DDIs, not all DDIs are clinically significant, and distinguishing between the clinically important and unimportant interactions can be challenging at times. And the clinical impact of DDI depends on various factors like the therapeutic index of drugs involved and the patient-specific variables. So all these factors could make the transporter-mediated DDIs challenging. Next slide, yeah. Revathi, you want to take this slide?
Sure. We do have our new launch of software, which is GPX. With this DDI interface, we have very competitive advantage compared to other softwares, where here in this using this interface, what we can do is we can model the effect of multiple substrates and multiple inhibitors at the same time. For example, you wanted to see how your compound is being affected by multiple inhibitors when administered at the same time. We can do that. That's doable with our GPX software. For example, here is a snapshot of some of the compounds. And you can see how we can select different compounds, like different substrates and different perpetrators, and model the effect of these perpetrators on this substrate. For example, in this case, it's a midazolam. So we can model the effect of one or two, or all together. We can go to any number of inhibitors and model the impact. Similarly, we also have with respect to metabolites, earlier there was a limitation like of one or two metabolites, but now now we can model the effect of multiple metabolites if we know that multiple metabolites are also inhibitors of specific enzyme or transporters. So this kind of helps to unravel the complex DDIs. For example, when there is induction or auto inhibition playing at the same point of time, so we can model both the effect of auto induction and time dependent inhibition at the same time if the compound has a potential for induction or or exerts time dependent inhibition. So here is a snapshot showing how the compound midazolam is affected by different perpetrators. Here, for example, inhibitors like strong inhibitors like ketoconazole, itraconazole, and its metabolite, and the effect of rifampicin, the inducer, and also the voriconazole 3A4 inhibitor. So we can see how different compounds are impacting based on whether they are administered together or given before or any point of time. So this is something which is new in our GPX, which is new in our software, which we have introduced through GPX.
Okay, so earlier we said that we will be providing the models to the users, and this is how you can access those model files and the reports for regulatory submission. So once you have logged into your Learn Upon account, you click on resources. And when it says archive, that is the databases like the GastroPlus modeling files for each DDI model that we have built in-house. And if you want to access the document or the report for that particular model, if you scroll down on the type, you have an option to select document, and this will show up all the PDF reports that we have included in this Learn Upon site for that particular database. So again, one thing I would like to highlight here is that we keep on updating this site with the models that we build regularly. And these are all the models that we have uploaded, and I think we have a couple of models that we will be uploading soon. And again, if you don't find the model that you are looking for, please feel free to contact us, and we can work on those models with developing and then providing you with the models based on the requests. And next slide, Revathi. So these slides just to highlight a couple of literature studies where the GastroPlus was used to perform the drug-drug interactions. Next slide. Yes, same additional literature on the GastroPlus DDI applications. Next slide. Yeah, at this time, I would like to take this opportunity to acknowledge Viera Lukachova, Grace, Joyce, Mike, Saima, and all the simulation studies and the technologies team who have equally contributed in building this whole house of the DDI network that we have.
All right, thank you, Revathi and Suvarchala. This brings us to the questions. Andy, we have a few questions you want to moderate the Q&A. Okay, well.
I think before we jump into the Q&A, I
Okay.
have a quick poll to launch. And it's basically just asking if you're interested in having someone from our team follow up with you, if you're interested in having a one-on-one with Susie. And then are you interested in participating in the University Plus program? I'm sorry, not one-on-one with Susie, but a 30-minute demonstration on how to use GastroPlus for DDIs. And then I'll give everybody a couple seconds to answer those questions.
Fantastic. Looks like we have some good interest in the demonstration, which we'd love to provide.
One more seconds here.
And then we'll have a few minutes for Q&A. I don't know that we'll get to all of them, but we can certainly follow up via email, put short meetings together, et cetera. So we will be sure to accommodate all of your questions, whether we can get to it in real time today or do follow-ups after the webinar.
All right, and I just ended the poll so we can take some questions.
All right, we'll start at the top. Robert Stratford asked, and I believe this was when Suvarchala was showing how to set up the perpetrators and transporters and legacy GastroPlus. The question is, do I need to have KM and Vmax for a 3A4 substrate to estimate FG? FG or FM, perhaps?
FG.
Is it FG or FM? If it is
Yeah.
FM, yes, we have an inbuilt converter within the GastroPlus to estimate the FM. But Revathi, yeah, please free to chime in and correct this.
So yeah, I mean, like for example, if you think that your compound is a 3A4 substrate, but you don't have the KM and Vmax parameter, instead you have the intrinsic clearance data or the in the recombinant enzymes or whatever you have in whichever in vitro system measured. Ideally, I mean, like the regulatory agencies prefer to have them characterize the in vitro kinetic parameters, both Vmax and Km in vitro, and use the data. But if we don't have the data, we can use the intrinsic clearance data from the in vitro systems and rely on admit predictor KM because this admit predictor that we have in-house is built using a vast library of compounds, and there are a quite number of spot-on predictions for KM that we had with many new investigational drugs that we worked for FIH predictions, where the KM, the admit predictor was very closer to what they have characterized in vitro. So in the absence of information, you can initially rely on the admit predictor KM and back calculate your Vmax based on the intrinsic clearance data.
Thank you very much. Let's do one more quick one before we have to take these offline, unfortunately. From Glenn Steventon, do you have models for OCT2, MATE, and MATE2K?
I think,
So
yeah, please go on. Yeah.
question, do we have models for OCT and MATES?
Yes.
Is that
That's the question,
okay.
yes.
Yes, I think we have metformin, which is a compound for OCT and substrate, and we have certain inhibitors of MATES like cimetidine in-house. So I think the validation process is complete. I think report is under construction also, but yeah, we do have models for OCT compounds.
Excellent. Thank you, Revathi. Thank you, Suvarchala. And I think that brings us to the top of the hour, maybe even a little bit older. So we will follow up with all remaining Q&A offline. Please do reach out to us if you'd like any follow-up conversations. Always happy to meet with you and put one-on-one meetings together. Thank you very much.
Thank you. We invite everyone to go to our website to learn more about DDI Risk Assessment. The webinar has been recorded for playback and will be available on our website and YouTube channel. This concludes our webinar for today. Thank you, everyone.
Thanks, everyone.